Om Swastiastu. Hello everyone, my name is Huliana Lim. I am the representative of Student Group Discussion B8 UDAYANA Medical School. In this occasion, we will going to present our student project of this clinical nutrition blog with the given topic Microbiome in IBS. And this is our group members, and the presentation will be continued by my peers. Thank you.
Hello everyone, my name is Gede Raka Samudra, number 0 to 3. Now I will explain to you about the chapter 1 introduction. 1.1 background. One of the most frequent functional gastrointestinal disorders seen in clinical practice is IBS or irritable bowel syndrome. It is a complex condition with a varied pathophysiology. Although current thinking suggests that IBS is complex in origin with still undetermined subgroups, the etiology of IBS remains unknown. As well as food hypersensitivity, it has been suggested that bad lifestyle, persistent infections, and a dysbiotic gut microbiome are to cause IBS symptoms in combination with predisposed factors. Despite the fact that irritable bowel syndrome affects a large percentage of the world's population, the pathogenesis pathways are poorly understood. Although a low-grade changes in the gut microbiome, there is discrepancy between studies and a consensus on functional gastrointestinal problems. The complex interaction of host variables, such as immunological activation, decreased epithelial barrier function and motility, etc., will be discussed later. Studies between IBS and the microbiome have revealed the importance of microbiome makeup, leading to the development of microbiome-based therapies. However, there is no definitive evidence that IBS can be treated. We also conducted a thorough analysis to look at the evidence supporting this correlation. 1.2 Problem formulation. The problem we are discussing in this student project include 1. What is the definition of microbiome in IBS? 2. What is the method of journal searching? 3. How is the digestion and absorption of microbiomes in IBS? 4. What is RTA? 5. What are the possible risks related to microbiome problems in IBS? 6. How does microbiome approach for IBS treatment? 1.3 Objectives. The objectives of this student project are 1. To understand the definition of microbiome and IBS. 2. To know the method of journal searching. 3. To understand digestion and absorption of microbiome in IBS. 4. To know the RTA. 5. To know the possible risks related to microbiome problems in IBS. 6. To find out how microbiome approach for IBS treatment. 1.4 Benefits. The benefits of this student project for us as medical students include complete knowledge of microbiome in IBS starting from its definition to the right treatment. Besides that, we can learn about digestion and absorption of microbiome in IBS, RTA, possible risks related to microbiome problems in IBS, and how microbiome approaches for IBS treatment. The benefit of scientific learning is that we can learn more about microbiomes in IBS and know how to treat it. The benefits for the community include increasing knowledge in the community who have a better understanding of microbiome and the appropriate therapy for it. That's all from me. Thank you.
Hello everyone, my name is Angelyne Mulia with last 3-digit number 051. So next I will explain about the definition of gut microbiome. Gut microbiome is defined as all microorganisms such as bacteria, viruses, fungi, and protozoa, and their genetic materials including genes and genomes that live in the human intestine. It contains approximately 30 trillion microbes and most of them are symbiotic with the host. Gut microbiome can affect multiple physiological functions such as metabolism, immunity, and inflammation. It interacts with epithelial cells and stromal cells to play various vital regulatory functions such as regulate barrier function and metabolism, maintain symbiosis and immune homeostasis, prevent pathogen infection, and control overgrowth of pathological organisms. That's all from me. Thank you.
Hello, my name is Azza with 3-digit student ID number 094. I will explain about the definition of irritable bowel syndrome or IBS. IBS is a functional gastrointestinal disorder characterized by recurrent episodes of abdominal discomfort associated with change in bowel habits. It is one of the most common GI disorders found in clinical practice. The pathogenesis of IBS is thought to result from abdominal brain-gut interaction, change in gut motility, visceral hypersensitivity, and other psychological stressors. Currently, The diagnosis of IBS was made according to a symptom-based classification system using the ROM4 criteria. That's all from me. Next.
Hello everybody, in this opportunity I want to explain the method of journal searching of our papers, Microbiome in IBS. First of all, let me introduce myself. My name is Putra and my student identification number is 200251104. Okay, so let's start. This paper uses literature review method, which the medical literatures are sought from PubMed, Google Scholar, ScienceDirect, Frontier, and PLOS ONE during the past 10 years from 2012 to 2022. The search keywords used were microbiome or gut microbiome, irritable bowel syndrome or IBS, probiotic, prebiotic, diet, risk factor, and treatment or therapy or management. These terms are combined with Boolean operators like and, or, and not to get specific international scientific journals. The search strategy generated 31 journals articles that were limited to English language publication. The data obtained were then arranged systematically according to the topic of the problem discussed, accompanied by inclusion and exclusion criteria. Okay, that's all the explanation of journal searching method. The presentation will be continued with my other friends. Thank you.
Hello everyone, my name is Gloria with last 3-digit number 162, and I'm going to explain about the digestion and absorption of microbiome. The gastrointestinal microbiota of the human body primarily has microbiomes consisting of bacteria. The large intestine, small intestine, and stomach are sites where bacteria reside, primarily in the large intestine. Bacteria that enters the body include commensal and pathogenic bacteria. Commensal bacteria are responsible for health benefit, whereas pathogenic colonization can lead to the development of the disease. The transmission route of microbiomes, digestion, and absorption differ according to the colonization strategy used inside the host. Several mechanisms are being used by bacteria to survive and transmit, for example, aerotolerance, viable but non-culturable dormancy, and sporulation. That mechanism is predominantly used by anaerobic bacteria. The source of transmission would be people, food, animal, and the built environment. When ingested by the host, the bacterium travels to the intestine. When the niche is uninhabited, bacteria can colonize. That's all from me. Thank you for listening, and the next slide will be presented by my friend. Thank you.
Thank you for the opportunity. My name is Krishna Putra Pramanda with student number 128. So now I would like to continue this presentation, still the same with the digestion and absorption of microbiome. In the progress of the digestion and absorption, the intestinal barrier will limit the entry of microbes into the intestinal lumen so that the homeostatic immune response is induced to maintain tolerance of the intestinal barrier to commensal microbes. When the intestinal barrier is penetrated by inflammatory mediators due to a pathogen that triggers an immune reaction, the inflammatory reaction occurs that affects the intestinal environment and changes the gut microbiota. Alteration in the gut microbiota may contribute to dysbiosis and the pathogenesis of IBS by altering gut immunity and integrity as well as modulating gut neuromuscular junction and brain access. Dysbiosis is a decrease or loss of gut microbial diversity due to changes in commensal bacteria into pathogens in human gut. I think that's all from me. This presentation will be continued by my friend. Thank you.
Hello everyone, my name is Natasya Pusparani with a 3-digit number of 227, and here I'll be presenting about recommended daily allowance or RDA. So intestinal microbiota plays a role in processing food metabolites digested by humans. Thus, it does correlate with dietary nutrients. A person's diet can change the microbiota gut profile, which possibly relates to dysregulation of intestinal permeability, visceral hypersensitivity, and its movement in IBS. Several dietary modulations can include high-fiber diets, a low-fermentable oligosaccharides, disaccharides, monosaccharides, and polyols, or FODMAP diet, low-inflammatory diets, and low-fat diets. Studies have shown a significant effect in limiting triggering foods that may cause major alterations of the gut microbiome or by having diets that can correct microbiota dysbiosis to help relieve symptoms of IBS.
Hello, my name is Luh Wina Saraswati with last 3 digits of student ID number 142. Now I will explain about the possible risks related to the microbiome problem in IBS. In several studies, it was found that the gut microbiota has an important role in several gastrointestinal diseases, including IBS. There are several pathogenesis-associated changes in the gut microbiota that may impact the development of IBS by altering the chains and integrity of the gut and modulating the gut neuromuscular junction and gut-brain axis. Some reports also state that the pathogenesis of IBS with dysbiosis, which is a condition that refers to a decrease or loss of microbial diversity and richness caused by the change in pathogenic commensal bacteria in the human gut. This dysbiosis state can lead to activation of the gut immune system and the potential for low-grade inflammation. In several cases of IBS, differences in the composition of the gut microbiota were found. In IBS patients, Bifidobacterium, Lactobacillus, and Faecalibacterium were found to be reduced, while regulators of Veillonella, Ruminicoccus, and pro-inflammatory bacterial species such as Enterobacteriaceae were increased. And lastly, there are several factors behind gastrointestinal disease related to the microbiome, including intestinal metabolism, intestinal permeability, and inflammatory pathway. That's all from me. The presentation will be continued by my friend. Thank you.
Hello everyone, my name is Huliana Lim. My student ID number is 20025111080. Now I will explain about the prebiotics that affects microbiome in a patient with IBS. Prebiotic defined as non-digestible food element in the human small intestine. Then it causes prebiotics to become a source of intestinal bacteria fermentation, which affect the host by explicitly increasing the growth and activity of bacteria, especially in patients with IBS, which is characterized by low microbacteria concentration. Based on clinical evidence, the type of prebiotics that have been discovered to have benefits in intestine, especially in IBS, are inulin type fructans and galacto-oligosaccharides. This can be found primarily in grains and vegetables. Prebiotic nutrition may be the first therapeutic choice for IBS patients, and the recommendation for prebiotic intake are not actually determined in guidelines. However, from the clinical trials shown that consuming 5 grams per day of fructooligosaccharides for about 6 weeks or 3.5 grams per day of galacto-oligosaccharides for about 12 weeks gives an improvement in IBS patient. Okay, I think that's it for the prebiotics. The presentation will be continued by my peers. Thank you.
Hello everyone, my name is Bagus with my number is 184. So, in the IBS conditions, there are treatments that can be offered to the patients. One of them is using prebiotics. Prebiotics is a living organism that can treat the IBS itself. It turns out that scientific evidence says that it is true that prebiotics have an indirect good effect to the IBS itself. There are prebiotic bacteria that have the most play role on the IBS, such as Lactobacillus, Bifidobacterium, and also Enterococcus. But there are other prebiotics bacteria that can give the same or the different effect to the IBS itself. As you can see on the right side of the table, there are many types of prebiotics that have a different outcome and a different effect to the IBS itself. Next.
Good morning or afternoon, doctor. My name is Felix with the series of student ID 099. We'll continue the presentation regarding the diets. So, most of patients with irritable bowel syndromes are usually modified their diets to specific types of food only to reduce their own symptoms. So, there are 3 types of trendy diets that are proven or influenced during the last few decades, which are first is the traditional dietary advice, the second is low fermentable oligosaccharides, disaccharides, monosaccharides, and polyol, all called FODMAPs, and also is gluten-free. So the first is the traditional dietary advice, which are considered the first line of the treatment. So the traditional dietary advice mainly does not require a specific type of or strict diet balance. It's only by reducing some certain type of food and also having a regular meal and adjusting fiber intake. The second is the low FODMAPs. which are mainly to reducing the intake of highly fermentable short chain and poorly absorbed carbohydrate, such as dairy-based meal, wheat, and based product. The food maps type of food are suspected to increases water secretion and fermentation in both small intestine and colon, which are causing a visceral hypersensitivity. Hence, by reducing the food maps food, it can reducing the meal trigger related symptoms. The last one is the gluten-free. The gluten are suspected to worsen some gastrointestinal symptoms in the patient, such as abdominal pain, bloating, stool consistency, and tiredness. So by reducing the gluten, it also reducing the symptoms that which shown on the patient. And also there are some types of diet, such as increasing the fiber and also many others. Thank you very much. Next.
Good afternoon, everyone. I'm Annisa Nuraini with the last 3 digits of NIM 110. I would like to continue the presentation on conclusion. So gut microbiome is defined as all microorganisms and their genetic materials, including genes and genomes that live in the human intestine. IBS, on the other hand, is a functional GI disorder marked by recurrent episodes of abdominal pain and change in bowel habits. This paper uses literature review method, which the medical literatures are sought from PubMed, Google Scholar, Science Direct, and Frontier during the past 10 years from 2012 to 2022. The GI microbiota of the human body primarily has biomass consisting of bacteria. Alterations in the gut microbiota may contribute to dysbiosis and the pathogenesis of IBS by altering the gut immunity and integrity. So a person's diet and choice of ingested food are able to change the profile of the gut from its normal state into dysregulation of the gut in IBS. There are several changes in the gut microbiota that may impact the development of IBS, which include the dysbiosis, which may lead to low-grade inflammation. Microbiome approach for IBS treatment includes the use of prebiotics, probiotics, and the regulation of diet for IBS patients, which can be traditional dietary advice, low FODMAP diet, or gluten-free diet.
Thank you for the presentation. And this is our references that we use in this student project. Alright, here it comes to the end. Thank you very much for all your attention. Sorry if we have many mistakes. I guess that's it. And thank you. Goodbye. Om Santi Santi Santi.